Using Clinov fragment as an enzyme of choice, we can proceed further with the sequencing after the availability of the reagents and buffers जिनकी हमें ज़रूरत पेश आती है during the experimental reactions. Deionized water, distilled water, ice cold as well as room temperature, optional nucleotide DATP, which is acceptable when we are going towards the procedural process, EDTA, and extension termination mixtures along with the chase mixture. Formamide loading buffer to stop the reactions, then TM buffer, that is trace magnesium chloride buffer, which is required. Enzymes में Clinov fragment from E. coli is going to be used. As this is part of the DNA polymerase 1, so this is the modified DNA polymerase 1, only Clinov fragment at a concentration of 5 units per microliter is available. We will need 2.5 units for each set of the 4 reaction mixtures. That is the 4 reaction mixtures of G, C, A and T reactions. Nucleic acids में the similar would be the situation that we will need normal DNTPs, we will need di-deoxynucleotide triphosphates. Normal DNTPs which are going to be used, they are prepared in stock solution of 1 millimolar concentration, whereas di-deoxynucleotide की concentration ज़्यादा होगी stock solutions की 5 millimolar concentration, which is prepared in water. Then we need primer. Oligonucleotide primer will be used, which is used at 0.5 picomolar concentration per microliter, and the enzyme is diluted in water again. Template would be the single-stranded DNA, and the concentration which is required to set up the sequencing reaction is 0.05 picomolar per microliter, that is equivalent to 0.15 microgram per microliter as far as quantity is concerned for the bacteriophage M13 single-stranded DNA. Radioactive ingredients और radioactive compounds में हम DATP यूज़ करेंगे. वेरिएंट में से कोई भी एक फॉर्म यूज़ की जा सकती है, अल्फा 32, अल्फा 33 for phosphate or alpha 35 for sulfur, or instead of using radioactive DATP, we can use the radio labeled oligonucleotide primer. Use of these radio labeled nucleotide, जैसे कि DATP की पर्पस ही यही है कि हम एक एंड जो है उसका न्यूली सिंथेसाइज्ड DNA स्ट्रैंड का लेबल कर सकें. The alternate option is we can start with radio labeled oligonucleotide primer. Microfuge tubes are required. Microfuge is required if the number of samples are very high, and we can start with the microtiter plate, 96 well plate, water bath preheated at 65 degrees centigrade. Centrifuges, the benchtop centrifuge, which can handle the 0.5 milliliter tubes, and the reaction mixture which we are going to use is shown in this slide, that is having different di-deoxynucleotide components along with their variable concentrations. Then chase in the given volume, DATP in the given volume. The normal nucleotide are listed here, and specific DDNTPs are listed here. Besides these, we are going to add trace CL, EDTA and water, and these are the volumes which we can use to make up the end volumes. So with these volumes and reaction mixtures, we can proceed further towards the sequencing. Once we prepare these kind of reaction mixtures, we can save them in a liquid form of 50 microliter and we can store them at minus 20 degrees centigrade.